Hi students, this is the third and final lecture on environmental health and toxicology. In this lecture, we will be learning about how organisms deal with exposure to toxic substances, which could include detoxification, cellular repair, excretion of toxic substances, or increased rates of cell replication. We will also be looking at the various ways that we measure the toxicity of different chemicals. In this lecture, we will also be looking at the difference between a chronic effect and an acute effect. And finally, we will look at the concept of risk assessment, which is how we evaluate the possible risk that exposure to a chemical may have on our health and development. Ultimately, this becomes a policy decision as to the best way to manage environmental risks. So, how does the body break down and excrete toxic substances? Well, most organisms have enzymes that process waste products and environmental poisons to reduce their toxicity. In mammals, most of these enzymes are located in the liver, the primary site of detoxification of both natural wastes and introduced poisons. The detoxification pathways in the liver function in a three-phase process. In phase one, a variety of enzymes act to introduce reactive and polar groups into the chemical. In phase two, the modified chemicals are conjugated with a charged species such as glutathione to create a more polar metabolite that can be more easily excreted by the body. In phase three, we further reduce the effects of waste products and environmental toxics by either additional metabolism or, more commonly, by eliminating them from the body through excretion. Volatile molecules such as carbon dioxide, hydrogen cyanide, and ketones are excreted via breathing. Some excess salts and other substances are excreted in sweat. Primarily, however, excretion is a function of the kidneys, which can eliminate significant amounts of soluble materials through urine formation. In this example, we see how alcohol is metabolized in the human body. Ethanol is metabolized to acetaldehyde by alcohol dehydrogenase, which is found in many tissues, including the gastric mucosa. Acetaldehyde is metabolized to acetate by acetaldehyde dehydrogenase, which is found predominantly in liver mitochondria. Acetate is then used by the muscle cells to produce acetyl-CoA using the enzyme acetyl-CoA synthase, and the acetyl-CoA is then used in the citric acid cycle. It takes roughly 90 minutes for a healthy liver to metabolize 30 mils, or 1 fluid US ounce, of pure ethanol, or approximately 42 minutes for a US standard drink, which is 14 grams or 14 mils or 0.47 US fluid ounces of pure ethanol. 1 hour is often used as a safety allowance. DNA damage due to environmental factors and normal metabolic processes inside the cell occurs at a rate of 1,000 to 1,000,000 molecular lesions per cell per day. A special enzyme, DNA ligase, shown here in color, encircles the double helix to repair a broken strand of DNA. DNA ligase is responsible for repairing the millions of DNA breaks generated during the normal course of a cell's life. Without molecules that can mend such breaks, cells can malfunction, die, or become cancerous. DNA ligases catalyze the crucial step of joining breaks in a duplex DNA during DNA repair, replication, and recombination, and require adenosine triphosphate ATP or nicotinamide adenine dinucleotide NAD as a cofactor. In this graphic, we see DNA ligase repairing chromosomal damage. The cells in tissues and organs that are regularly exposed to toxic or irritating substances and physical wear and tear are equipped with mechanisms to repair damage. This includes high cellular reproductive rates to replace the injured or damaged cells. This is common in the skin and the epithelial linings of the gastrointestinal tract, blood vessels, lungs, and the urogenital system. As replacement continues, however, the chances for mutation and error in the repair process increases. Thus, these areas are also highly prone to tumor formation and cancer. According to the National Cancer Institute, among the 20 most common cancers in 2016 are bladder cancer, colon and rectal cancer, endometrial cancer, kidney cancer, liver cancer, and lung cancer, all of which are located in parts of the body that are regularly exposed to toxic substances. So how do we know if a chemical is toxic? What is a safe dose? How do we go about measuring toxicity? In 1540, the Swiss scientist Paracelsus said, "The dose makes the poison," by which he means that almost everything is toxic at very high levels, but can be safe if diluted enough. This remains the most basic principle of toxicology. Sodium chloride, or table salt, for instance, is essential for human life in small doses. If you were to be forced to eat a kilogram of salt all at once, however, it would make you very sick. A similar amount injected into your bloodstream would be lethal. How a material is delivered, at what rate, through which route of entry, and in what medium plays a vital role in determining toxicity. The most commonly used and widely accepted toxicity test is to expose a population of laboratory animals to measured doses of a specific substance under controlled conditions. This procedure is expensive, time-consuming, and often painful and debilitating to the animals being tested. It commonly takes hundreds or even thousands of animals, several years of hard work, and hundreds of thousands of dollars to thoroughly test the effects of a toxic at very low doses. Because it's so time-consuming, many chemicals are never tested at all. More humane toxicity tests using computer simulations of model reactions, cell cultures, and other substitutes for whole living animals are being developed. However, conventional large-scale animal testing is the method in which scientists have the most confidence and on which most public policies about pollution and environmental or occupational health hazards are based. The figure shows a typical dose-response curve for exposure to the metal selenium. Some individuals are very sensitive to the material, while others are insensitive. Most, however, fall in a middle category, forming a bell-shaped curve. The question for regulators and politicians is whether we should set pollution levels that will protect everyone, including the most sensitive people, or only aim to protect the average person. It might cost billions of extra dollars to protect a very small number of individuals at the extreme end of the curve. What do you think? Is that a good use of resources? Dose-response curves aren't always symmetrical, making it difficult to compare toxicity of unlike chemicals or different species of organisms. A convenient way to describe toxicity of a chemical is to determine the dose to which 50% of the test population is sensitive. In the case of a lethal dose, LD, this is called the LD50. Unrelated species can react very differently to the same poison, not only because body and metabolism differ. Even closely related species can have very dissimilar reactions to a particular chemical. Hamsters, for instance, are nearly 5,000 times less sensitive to some dioxins than are guinea pigs. Of 226 chemicals found to be carcinogenic in either rats or mice, 95 caused cancer in one species but not the other. These variations make it difficult to estimate the risks for humans because we don't consider it ethical to perform controlled experiments in which we deliberately expose people to toxins. In this figure, we see lethal doses of some common chemicals. As Paracelsus noted, everything is a poison. It just depends on the dose. For example, drinking about 6 liters of water, 118 cups of coffee, or 13 shots of alcohol would deliver a lethal dose for most humans. In this table, the least toxic substance is water. The most toxic is a toxin from the bacterium Bacillus botulinum. Botulinum is currently used as a cosmetic treatment to remove wrinkles in the skin, but it is also a deadly poison. Special care must be taken when canning food at home to ensure botulinum does not enter the food. Most of the toxic effects that have we discussed so far have been acute effects. That is, they are caused by a single exposure to the chemical and result in an immediate health crisis. Often, if the individual experiencing an acute reaction survives this immediate crisis, the effects are reversible. Chronic effects, on the other hand, are long lasting, perhaps even permanent. A chronic effect can result from a single dose of a very toxic substance, or it can be the result of a continuous or repeated, some lethal exposure. We also describe long lasting exposures as chronic, although their effects may or may not persist after the toxic agent is removed. It usually is difficult to assess the specific health risks of chronic exposures because other factors such as aging or normal diseases act simultaneously with the factor under study. It often requires very large populations of experimental animals to obtain statistically significant results for low-level chronic exposures. One of the most challenging issues in toxicology is to predict the effect of a chemical at very, very low doses. This graph shows three possible results from low doses of a toxic chemical. Curve A shows a baseline level of response in the population, even at zero dose. This suggests that some factor in the environment also causes this response. Curve B shows a straight line relationship from the highest dose to zero exposure. Many carcinogens and mutagens show this kind of response. Any exposure to such agents, no matter how small, carries some risk. Curve C shows a threshold for the exposure where some minimal dose is necessary before any effect can be observed. This generally suggests the presence of a defensive mechanism that prevents the toxin from reaching its target in an active form or repairs the damage that the toxic causes. Low levels of exposure to the material in question may have no deleterious effects, and it might not be necessary to try to keep exposures to a minimum. As science advances, our ability to detect lower and lower concentrations of chemicals becomes better. Toxic materials may seem to be more widespread now than in the past, and this is surely a valid perception for many substances. The daily reports we hear of new chemicals found in new places, however, are also due in part to our more sensitive measuring techniques. 20 years ago, parts per million were generally the limits of detection for most materials. Anything below that amount was often reported as zero or absent, rather than more accurately as undetected. A decade ago, new machines and techniques were developed to measure parts per billion. Suddenly, new substances were found where none had been suspected. Now we can detect parts per trillion or even parts per quadrillion in some cases. The increasingly sophisticated measuring capabilities may lead us to believe that toxic materials have become more prevalent. In fact, our environment may be no more dangerous. We're just better at finding trace amounts. A complication in assessing risk is that the effects of low doses of some toxics and health hazards can be nonlinear. They may be either more or less dangerous than would be predicted from exposure to higher doses. For example, low doses of DHEP suppress activity of an enzyme essential for rat brain development. This is surprising because higher doses stimulated this enzyme. Thus, low doses can be more damaging to brain development than expected. On the other hand, very low amounts of radiation can seem to be protective against certain cancers. This is perplexing because ionizing radiation has long been recognized as a human carcinogen. It's thought now, however, that very low radiation exposure may stimulate DNA repair along with enzymes that destroy free radicals. Those are atoms with unpaired reactive electrons in their outer shells. Activating these repair mechanisms may defend us from other unrelated hazards. These nonlinear effects are called hormesis. Even if we know with some certainty how toxic a specific chemical is in laboratory tests, it's still difficult to determine risk. the probability of harm multiplied by the probability of exposure. If that chemical is released into the environment. As we have seen, many factors complicate the movement and fate of chemicals both around us and within our bodies. Furthermore, public perception of relative dangers from environmental hazards can be skewed so that some risks seem much more important than others. For instance, which is more risky for you, putting your hand into the open mouth of an alligator or drinking a glass of tap water? How would we even determine the process of calculating the risk? People with social, political, or economic interests, including environmentalists, tend to downplay certain risks and emphasize others that suit their own agendas. We do this individually as well, building up the dangers of things that don't benefit us while diminishing or ignoring the negative aspects of activities we enjoy or profit from. Most people have difficulty understanding and believing probabilities. We feel that there must be patterns and connections in events, even though statistical theory says otherwise. If the coin turned up heads last time, we feel certain that it will turn up tails next time. In the same way, it is difficult to understand the meaning of a 1 in 10,000 risk of being poisoned by a chemical. We tend to have an irrational fear or distrust of certain technologies or activities that lead us to overestimate their dangers. Nuclear power, for instance, is viewed as very risky, while coal-burning power plants seem to be familiar and relatively benign. In fact, coal mining, shipping, and combustion cause an estimated 10,000 deaths each year in the United States, compared with none known so far for nuclear power generation. An old, familiar technology seems safer and more acceptable than does a new, unknown one. Alarmist myths and fallacies spread through society, often fueled by xenophobia, politics, or religion. For example, the World Health Organization campaign to eradicate polio worldwide has been thwarted by religious leaders in northern Nigeria, the last country where the disease remains widespread. They claim that oral vaccination is a U.S. plot to spread AIDS or infertility among Muslims. Our personal experiences often are misleading. When we have not personally experienced a bad outcome, we feel it's more rare and unlikely to occur than it actually may be. Furthermore, the anxieties generated by life's gambles make us want to deny uncertainty and to misjudge many risks. We have an exaggerated view of our own abilities to control our fate. We generally consider ourselves above average drivers, safer than most when using appliances or power tools, and less likely than others to suffer medical problems such as heart attacks. People often feel they can avoid hazards because they are wiser or luckier than others. The news media give us a biased perspective as well on the frequency of certain kinds of health hazards, overreporting some accidents or diseases while downplaying or underreporting others. Sensational, gory, or especially frightful causes of death, such as murders, plane crashes, fires, or terrible accidents, receive a disproportionate amount of attention in the public media. Heart disease, cancer, and stroke kill nearly 15 times as many people in the United States as do accidents, and 75 times as many people as do homicides. But the emphasis placed by the media on accidents and homicides is nearly inversely proportional to the relative frequency, compared with either cardiovascular disease or cancer. This gives us an inaccurate picture of the real risk to which we are exposed. For instance, how dangerous is trick skating? Many parents regard them as extremely risky, while many students, especially males, believe the risks are acceptable. Perhaps the more important question is whether the benefits outweigh the risks. How much is it worth to minimize and avoid exposure to certain risks? Most people will tolerate a higher probability of occurrence of an event if the harm caused by that event is low. Conversely, harm of greater severity is acceptable only at low levels of frequency. A 1 in 10,000 chance of being killed might be of more concern to you than a 1 in 100 chance of being injured. For most people, a 1 in 100,000 chance of dying from some event or some factor is a threshold for changing what they do. That is, If the chance of death is less than 1 in 100,000, we are not likely to be worried enough to change our ways. If the risk is greater, we will probably do something about it. The Environmental Protection Agency generally assumes that a risk of 1 in 1 million is acceptable for most environmental hazards. Critics of this policy ask, acceptable to whom? For activities that we enjoy or find profitable, we are often willing to accept far greater risks than this general threshold. Conversely, for risks that benefit someone else, we demand far higher protection. For instance, your chance of dying in a motor vehicle accident in any given year are about 1 in 5,000, but that doesn't deter many people from riding in automobiles. Your chances of dying from lung cancer if you smoke 1 pack of cigarettes per day are about 1 in 1,000. By comparison, the risk from drinking water with the EPA limit of trichloroethylene is about 2 in 1 billion. Strangely, many people demand water with 0 levels of trichloroethylene while continuing to smoke cigarettes. This chart shows the relative risk of various activities. Humans have a 50% chance of dying from heart disease, but only a 1 in a million chance of dying from botulism. The biggest problem in making regulatory decisions is that we are usually exposed to many sources of harm, often unknowingly. It's difficult to separate the effects of all these different hazards and to evaluate their risks accurately, especially when the exposures are near the threshold of measurement and response. In spite of often vague and contradictory data, public policymakers must make decisions. The case of the sweetener saccharin is a good example of the complexities and uncertainties of risk assessment in public health. Studies in the 1970s at the University of Wisconsin and the Canadian Health Protection Branch suggested a link between saccharin and bladder cancer in male rats. Critics of these studies pointed out that humans would have to drink 800 cans of diet soda per day to get a saccharin dose equivalent to that given to the rats. Furthermore, they argued this response may be unique to male rats. In 2000, the U.S. Department of Health concluded a study that found no association between saccharin and cancer in humans. Congress ordered that all warnings be removed from saccharin-containing products. Risk assessment consists of an objective evaluation of risk, in which assumptions and uncertainties are clearly considered and presented. Part of the difficulty in risk management is that both the quantities by which risk assessment is concerned, the potential loss and probability of occurrence, can be very difficult to measure. The chance of error in measuring these two concepts is high. In ideal risk management, a prioritization process is followed whereby the risks with the greatest loss or impact and the greatest probability of occurring are handled first, and the risks with lower probability of occurrence and lower loss are handled in descending order. In practice, the process of assessing overall risk can be difficult, and balancing resources used to mitigate between risks with a high probability of occurrence but lower loss versus a risk with high loss but lower probability of occurrence can often be mishandled. In this lecture, we have learned about how the body uses enzymes, mostly located in the liver, to detoxify chemical agents that enter our body. Our cells can repair damage to DNA through complex systems of cellular repair. In locations where cells are exposed to a high frequency of agents, it replaces cells more frequently. We test chemical and biological agents on animals to help determine the doses of these agents that are either safe or harmful, and from these experiments we develop dose-response curves. A common dose-response curve is the LD50, which helps us determine the dose at which 50% of the population will die. We also learned about the difference between acute effects and chronic effects. Acute exposure is usually a single exposure to an agent and may be very dangerous depending on the dose and route of exposure. A chronic effect can result from a single exposure or a long-term exposure. We also learned about risk assessment and risk management. Risk assessment is about identifying and analyzing risks. Risk management is about prioritizing the risk and developing a response. In conclusion, humans have made great progress reducing some of the worst diseases that have long plagued humans, such as smallpox, measles, diphtheria, tetanus, and microbial infections. Even though we are exposed to numerous chemical compounds every day, infectious diseases are still the most common source of mortality and morbidity. Unfortunately, the chronic conditions, which were once typical of rich countries, are now becoming problems nearly everywhere. These chronic conditions include heart disease, depression, diabetes, and cancer associated with smoking, liver disease associated with alcohol consumption, and many others. On top of this chronic disease burden, we now have many new emergent diseases that have an extra disease burden to the developing world. These new emergent diseases are also quite likely to arrive in developed countries such as the United States and Europe because of the ease of travel. Our industrial society introduces thousands of new chemicals every year. However, the effects of these chemicals on human and environmental health remains unclear. Many of these chemicals are not widely studied, and therefore toxicity and toxicology data are largely unknown.